attend, but Ye is going to cover for him. So this talk is going to be about finding differential interactions in high-C data. And Ye is also from University of Wisconsin-Madison from Tündüz Gelişis group.
Thanks, Fahad, for introduction. So I'm going to present TreeHiC. It is utilizing a hierarchical test for chromatin differential interaction. And we will use HiC as the illustration. So to detect the differential chromatin interaction, basically we have HiC contact matrix under one condition, under like multiple condition, like or different tissue or cell line, and we want to know which interaction, like which interaction from one region to another region that is enriched, and one condition but not enriched has no like interaction signal, and another condition. And we are aware that there are some ad hoc procedure or some model exists to do such differential interaction. However, there are some major problem. For example, some models that work well under low resolution because the read coverage is relatively high. However, for high resolution, the contact matrix will be sparse and the signal to noise ratio will be low. So the detection power can be a potential problem. And also for the high resolution, there will be a large number of hypothesis tests. So when we do Benjamin-Hochberg procedure to control the FDR, it may fail because the most important assumption for Benjamin-Hochberg procedure is that all the hypotheses are independent. So for large scale hypothesis, and especially for high-C because of spatial dependency, such assumption will not hold. So eventually, the force discovery rate cannot be controlled. Force discovery here is a ratio of round rejection among all the rejection. And also the model that can be only applied to multiple replicates because you need multiple replicates to estimate the variance or sparsity. So it cannot be applied to one replicate. Therefore, for TreeHiC, our target is to detect the differential interaction while maintain high detection power and control FDR at the same time. And also it can be applicable to high resolution and low resolution with different sparsity, different signal to noise ratio. And also it can be applied to one replicate as well. So there are three major steps for TreeHiC. The first one is partition and extreme mode search. So first we get contact matrix for different condition. We first take average of all the biological replicate high-C contact and divided by another average contact count matrix. And so later on, the model will work on this like still asymmetric matrix, but each cell will be log2 fold change of contact count between two conditions. So this is like log2 fold change contact matrix we internally get. And instead of testing on all the cells in the contact matrix, we will do a partition and only test the extrema, meaning the maximum and minimum within one partition. In other words, if the maximum and minimum fail to get rejected, they are failed to be considered as differential interaction. There's no need to go further and test those sub-maximum and sub-minimum. So the number of hypothesis tests will be reduced significantly. Then how can we partition the the log2 fold change contact matrix? We'll utilize a more small decomposition. It is a well-established algorithm in topology, and there's a parameter called persistency. We can use it to tune how further we will partition the log2 fold change contact matrix. And here is an illustration. If we start with persistence as 1, there's no partition. And we go further down, there will be a few more partitions. And to be noted that the further partition will be like nested within the parent segment. So it just like go further down to partition. And eventually we can use persistence to control like how many partitions we will get. Here is a 3D illustration. Like first we will start with a plateau to get the first persistent parameter and get the first like partition, and we will test the maximum that is a green dot and the minimum that is a blue dot here. And if they are rejected, we'll go further and partition more using like another persistent parameter. So as you can see, all the tasks are nested within this like parent segment. So it's natural to utilize a hierarchical testing for the task. It is proposed by statisticians like 10 years ago, and the idea is that like when we do the first partition, we'll put all the maximum, minimum of the first partition on the first level of the hypothesis testing tree. And if it is rejected, that becomes green, and we will go further and partition this parent segment and put the further partition maximum and minimum on the second level of the hypothesis tree. And we also will apply Benjamin Hochberg procedure within one family, meaning that they share the same parent node. So within one family, we can control FDR at the alpha level. And the statistician has theoretically proved that the overall FDR can be controlled at this level. And alpha is like Benjamin Hochberg procedure FDR threshold you want to set. And the third procedure is to get the p-value, because the p-value, how we obtain the p-value is independent from how we construct the tree, because the tree is dependent on how we partition the contact matrix. So the p-value can be obtained by like existing model C, DIFHiC, or we can use permutation. Basically, the idea for permutation is to permute all the log2 fold chain of all the cells that share the same genomic distance and get the probability for observing such log2 fold change under such genomic distance. And we carry out two simulations starting. First, we use HiC compare to simulate one replicate under each condition. And we compare with DIFHiC permutation and TreeDIFHiC, which utilize hierarchical testing structure using DIFHiC p-value and tree permutation. And basically, we simulate under resolution that is dense, and also the signal to noise ratio being small, medium, and large. And we want to have the FDR, the empirical FDR, to be controlled under the dashed line. And also we want to have like high sensitivity and specificity as high as possible. And as you can see, like only using TreeHiC, we can control the FDR under the dashed line. And also we simulated a sparse HiC contact matrix of different signal to noise ratio, and still using TreeHiC we can control the FDR and also achieve as high sensitivity and specificity as high as possible. And the second simulation we simulate using find. There will be a specific talk about find this afternoon. And we simulate two replicate under each condition and compare with DIFHiC find and using DIFHiC p-value and tree structure, basically TreeDIFHiC and tree permutation. And again, like using the TreeHiC, we can control the FDR and achieve decent specificity and sensitivity. Okay, so those are about simulation. For real data, we want to see whether detection power is high enough. So we do the differential interaction for K562 and GM cell line, and we use the detection using 4 replicates as gold standard and try to recover using replicates that is 2 or 3 under each condition. So basically, we want to achieve as high recovery proportion as possible. So as you can see here, the TreeDIFHiC, basically DIFHiC p-value using tree hierarchical testing structure achieve the highest recovery proportion, meaning highest detection power. And also we want to know, like, we have high detection power, whether we can control the FDR. So we do the differential interaction within one condition, meaning like we compare 2 replicates and of K562 and another 2 replicate of 2562. So basically we want to have like as low as possible of the differential interaction. So as you can see, the TreeHiC achieved the lowest force discovery rate. And also we overlap the differential interaction with the differential express strings, and again, TreeHiC can achieve like high overlapping proportion with the differential express strings. So basically, maybe it has some biological support of it. that is all about TreeHiC we want to present, and the pipeline is now available on GitHub. So thank you so much. I'd like to take questions.
Just one quick question. So you mentioned that you investigated the low distribution and high, for example, when the high. But did you also try to compare sample when there is one low versus high? And does it allow this possibility to do this really when there is different samples to compare?
Do you mean like compare? Sorry, like do you mean like compare signal that is low and one condition signal
yes,
is high?
exactly.
Yeah, it is considered within this model because at first we will get a log2 fold change contact matrix. So it can be maximum or can be minimum of the log2 fold change.
Oh, thanks.
Okay, thank you.
We're setting up here, but our this morning session